Dear students, Assalamu alaikum. In the last lecture, we studied about the glomerular filtration, its rate, and some of its determinants. The objective of today's lecture are to learn about glomerular capillary hydrostatic pressure and physiological control, autoregulation, and myogenic autoregulation of glomerular filtration rate and renal blood flow. In the last lecture, we studied three determinants of glomerular filtration rate. We'll continue with the fourth. Determinant number 4, glomerular capillary hydrostatic pressure, Pg. It has been estimated to be about 60 millimeter of mercury under normal conditions. Now keep in mind that it is the changes in glomerular capillary hydrostatic pressure which serve as the primary means for the physiological regulation of glomerular filtration rate. Glomerular hydrostatic pressure is determined by three variables, each of which is under separate physiological controls. Number 1, arterial pressure. Number 2, afferent arteriolar resistance. And number 3, efferent arteriolar resistance. Increased arterial pressure raises the glomerular hydrostatic pressure and increases the glomerular filtration rate. But this effect is buffered by autoregulatory mechanisms, and therefore arterial pressure has not that much control. The second is the afferent arteriolar resistance. Now, increased resistance of afferent arterioles, as you can see in the opposite figure, reduces the glomerular hydrostatic pressure and therefore decreases the glomerular filtration rate. And consequently, the dilation of the afferent arteriole increases both glomerular hydrostatic pressure and glomerular filtration rate. Constriction of the efferent arterioles increases the resistance to outflow from the glomerular capillaries, and this raises the glomerular hydrostatic pressure, and the glomerular filtration increases slightly. Now, this efferent arterial constriction has a biphasic effect on glomerular filtration rate. At moderate levels of constriction, there is a slight increase in glomerular filtration rate, but with severe constriction, there is a decrease in glomerular filtration rate. Now let's see how that occurs. The decrease in glomerular filtration rate by severe constriction occurs as the efferent constriction becomes severe, and as the plasma protein concentration increases, there is a rapid increase in colloid osmotic pressure due to Donan's effect. Now, this increase in glomerular capillary colloid osmotic pressure decreases the glomerular filtration rate. Increase in the glomerular hydrostatic pressure raise the glomerular filtration rate, while the decreases in the glomerular hydrostatic pressure reduce the glomerular filtration rate. So that is the summary of what we have just studied. Now, let's revise some of the factors that decrease the glomerular filtration rate. You can see in the first row that a decrease in the glomerular capillary filtration coefficient causes a decrease in glomerular filtration rate, and that may occur because of some renal disease, diabetes mellitus, or hypertension. And secondly, in the second row, an increase in the Bowman capsule hydrostatic pressure, PB, also leads to a decrease in glomerular filtration rate because of urinary tract obstruction, like kidney stones or urethral stones. Then an increase in the colloid osmotic pressure of glomerular capillary plasma proteins also lead to a decrease in the glomerular filtration rate, and this may happen because of a decrease in the renal blood flow or increased plasma proteins. Now, a decrease in the glomerular capillary hydrostatic pressure, as we have just studied in the last couple of slides, that causes a decrease in the glomerular filtration rate, and this may be because of a decrease in the arterial pressure or a decrease in the efferent arteriolar resistance, or an increase in the afferent arterial resistance. Now, in case of a decreased arterial pressure, this has only a small effect because of the autoregulation of the arterial pressure, while a decrease in efferent arteriolar resistance is because of decreased angiotensin II formation. If the drugs that block the angiotensin II formation are being used. And lastly, an increase in the afferent arteriolar resistance. This may occur because of increased sympathetic activity or due to the vasoconstrictor hormones like norepinephrine or endothelin, which we will study a little later in the lecture also. So this summarizes the major factors that can cause a decrease in the glomerular filtration rate. The table is given in your textbook of Guyton, so do study it in detail and remember these factors. The next topic is renal blood flow. The combined blood flow through both kidneys is about 1100 milliliters per minute, and it's about 22% of the cardiac output. The kidneys consume oxygen at twice the rate of the brain, and a large fraction of this oxygen consumption is related to the active sodium reabsorption by the renal tubules. Now, the renal blood flow is determined by the difference between the renal artery pressure and the renal vein pressure divided by the total renal vascular resistance. The renal artery pressure is equal to the systemic arterial pressure. The renal vein pressure averages 3 to 4 millimeters of mercury. And the total renal vascular resistance, like in other vascular beds, is equal to the sum of resistances in smaller renal arteries, arterioles, capillaries, and veins. Most of the renal vascular resistance is in three major segments: interlobular arteries, afferent arterioles, and efferent arterioles. And the resistance of these vessels is controlled by sympathetic nervous system, hormones, and the local internal renal control mechanisms. We'll study these mechanisms later in the lecture. An increase or decrease in the vascular resistance of the segments of kidneys tends to reduce or increase the renal blood flow respectively, if the renal artery and renal vein pressures remain constant. The renal cortex receives most of the kidney's blood flow, while blood flow in the renal medulla accounts for only 1 to 2% of the total renal blood flow. As we learned in a previous lecture, blood to the medulla is supplied by a specialized portion of the peritubular capillary system called vasa recta. Vasa recta descend into the medulla and return to the cortex before emptying into the nervous system. Emptying into the venous system. The determinants of glomerular filtration rate that are most variable and subject to physiological control include the glomerular hydrostatic pressure and the glomerular capillary colloid osmotic pressure. And these variables, in turn, are influenced by the sympathetic nervous system, hormones, and autacoids. Strong activation of the renal sympathetic nerves can constrict the renal arterioles and decrease renal blood flow and glomerular filtration rate. And these renal sympathetic nerves are most important in reducing the glomerular filtration rate during severe hemorrhage. Coming to the hormonal and the Autacoid control, norepinephrine, epinephrine, and endothelin, they constrict the renal blood vessels and decrease the glomerular filtration rate. Endothelin is a peptide released by damaged vascular endothelial cells of the kidneys. Angiotensin. Now, although the receptors of angiotensin are present in all the blood vessels of the kidneys, there's some peculiarities about its action regarding the afferent arterioles and the efferent arterioles. The afferent arterioles are protected from the angiotensin II mediated constriction due to the release of vasodilators which counteract effects of angiotensin II in these blood vessels. On the other hand, the efferent arterioles are highly sensitive to the angiotensin II. Therefore, angiotensin II preferentially constricts the efferent arterioles. It prevents decreases in glomerular hydrostatic pressure and the glomerular filtration rate. And it increases the tubular reabsorption of sodium and water, which restores the blood volume and the blood pressure. Endothelial derived nitric oxide. It decreases the renal vascular resistance and increases the glomerular filtration rate. Prostaglandins and bradykinin decrease the renal vascular resistance and tend to increase the glomerular filtration rate. These substances are not of importance in regulating the renal blood flow or the glomerular filtration rate in normal conditions, but they may decrease the vasoconstrictor effects of sympathetic nerves or angiotensin II on the afferent arterioles, an effect that I mentioned just a couple of slides earlier. Coming now to the autoregulation of the glomerular filtration rate and the renal blood flow. Feedback mechanisms intrinsic to the kidneys keep the renal blood flow and the glomerular filtration rate constant, despite changes in the arterial blood pressure. Now, this constancy of glomerular filtration rate and renal blood flow is referred to as the autoregulation. In general, the renal blood flow is autoregulated in parallel with the glomerular filtration rate, but it is the glomerular filtration rate which is more efficiently autoregulated under certain conditions. The kidneys have a feedback mechanism that links. It's important to understand that this feedback mechanism links changes in the sodium chloride concentration at the macula densa of the distal tubule with the control of renal arterial resistance and the autoregulation of glomerular filtration rate. So mostly this feedback autoregulates renal blood flow and the GFR in parallel, and this feedback is called tubuloglomerular Tubuloglomerular Feedback. Tubuloglomerular Feedback. The tubuloglomerular feedback mechanism has two components to control the glomerular filtration rate. And number one, an afferent arteriolar feedback mechanism, and number two, an efferent arteriolar feedback mechanism. These feedback mechanisms depend on special anatomical arrangements of the juxtaglomerular complex. The juxtaglomerular complex, if you can remember from the previous lecture, consists of number one, macular denser cells, which are present in the initial portion of the distal tubule, and the juxtaglomerular cells, which are present in the walls of the afferent and efferent arterioles. The macula densa is a specialized group of epithelial cells in the distal tubules that comes in close contact with the afferent and efferent arterioles. This figure depicts the structure of the juxtaglomerular apparatus, showing the juxtaglomerular cells in the afferent and efferent arterioles and the macula densa cells in the distal tubule. The tubuloglomerular feedback functions as follows. The macula densa cells sense changes in the fluid volume delivered to the distal tubule, and a decreased glomerular filtration rate slows the flow rate in the loop of Henle, causes increased reabsorption of sodium and chloride ions in the ascending loop of Henle, and this reduces the concentration of sodium chloride at the macula densa cells. Now, this decrease in sodium chloride concentration at the macula densa has two effects. Number one, it decreases resistance to blood flow in the afferent arterioles. And as we learned earlier, this raises the glomerular hydrostatic pressure and helps return glomerular filtration rate toward normal. The second effect is that it increases the renin release from the juxtaglomerular cells of the afferent and efferent arterioles, which are the major storage sites for the renin. Renin functions as an enzyme to increase the formation of angiotensin 1, which is converted to angiotensin 2. And finally, angiotensin 2 constricts the efferent arterioles, increasing the glomerular hydrostatic pressure and returning the glomerular filtration rate towards normal. This is a very important diagram showing the macula densa feedback mechanism for autoregulation of glomerular hydrostatic pressure and glomerular filtration rate during decreased arterial pressure. Therefore, the summary of tubuloglomerular feedback is that a decreased macula densa sodium chloride causes dilation of afferent arterioles and increased renin release, leading to formation of angiotensin II, and this angiotensin II constricts the efferent arterioles, increasing the glomerular hydrostatic pressure and helping the glomerular filtration rate to return toward normal. The next topic is the myogenic autoregulation of renal blood flow and the GFR. Myogenic autoregulation is another mechanism that contributes to the maintenance of a constant renal blood flow and the glomerular filtration rate. Myogenic mechanism is the ability of individual blood vessels to resist stretching during increased arterial pressure. And this vascular wall stretch allows increased movement of calcium ions into the cells, causing them to contract. And this contraction prevents excessive stretch of the vessel, and by raising vascular resistance, it prevents excessive increases in renal blood flow and glomerular filtration rate when the arterial pressure increases. Effect of high protein intake and increased blood glucose on the renal blood flow and the glomerular filtration rate. Although renal blood flow and glomerular filtration rate are relatively stable under most conditions, there are circumstances in which they change significantly. For example, high protein intake increases both renal blood flow and the glomerular filtration rate. Now let's see how that happens. This figure gives a likely explanation for the increased glomerular filtration rate. A high protein meal increases the release of amino acids in the blood, which are reabsorbed in the proximal tubule. Because amino acids and sodium are reabsorbed together by the proximal tubules, increased amino acid reabsorption also stimulates the sodium reabsorption in the proximal tubules. So this reabsorption of sodium decreases the sodium delivery to the macula densa, which then causes a decrease in afferent arteriolar resistance by the tubuloglomerular feedback mechanism that we just studied. And the decreased afferent arterial resistance then raises the renal blood flow and the glomerular filtration rate back to the normal. Now, as to the effect of increased blood glucose, a similar mechanism may also explain the increases in the renal blood flow and the glomerular filtration rate that occur with increased blood glucose levels in the uncontrolled diabetes mellitus. Now, this concludes our discussion about the glomerular filtration, which is the first step in the formation of urine. We will continue with the study of the next step, which is the tubular reabsorption, in the next lecture.